Hello everybody, welcome to the webinar today for DNA cell cycle analysis with Multicycle in FCS Express. Today we have Kevin Becker on the line from Phoenix Flow Systems, as well as myself, Sean Burke from Novo Software. Novo Software and Phoenix Flow Systems have both been in business for over 15 years now, and we have partnered on many projects in the past. And Novo Software benefits from Phoenix Flow Systems' vast knowledge in the area of flow cytometry and their many industry relationships, while Phoenix Flow Systems has benefited from our ability to quickly create custom professional solutions in the area of flow cytometry data analysis. And currently, Novo Software and Phoenix Flow Systems have collaborated to integrate the highly successful DNA cell cycle analysis application MultiCycle into our flagship project FCS Express. The basic version of MultiCycle is included for free with FCS Express. This basic version works for normally distributed data with one cell cycle, and it does not take into account any noise or background populations. However, the full version of MultiCycle can be purchased at any time from Novo Software. Cell cycle data in FCS Express is going to work for flow cytometry data or image cytometry data. So it's a great tool for working with any of the kind of data acquisition platforms that you're using. And today we're going to get started by going over some of the basics of cell cycle data, some of the theory, and how to insert multi-cycle plots within FCS Express. We'll then continue on in the webinar with Kevin Becker giving us a more detailed overview of the MultiCycle platform and how all the different features and functions within that work and can apply to your cell cycle data. So to get started, let's have a basic introduction to cell cycle analysis, talk about the theory and data collection a little bit. So you can see here in our diagram, we have a diagram of the cell cycle of cells progressing from G0 to G1 on into S phase, G2, and M. And you can see that as the cells progress through the cell cycle, the amount of DNA changes from 2N to 4N in a very normal manner. When a DNA stain is used, or when DNA is stained using a variety of dyes, the fluorescent intensity of those dyes are directly proportional to the amount of DNA. This allows you to define G1 and G2 peaks in data, as well as all the variable amounts in the S phase. And when you look at a theoretical model of DNA analysis, you see that there's peaks with discrete 2N and 4N populations. And mathematical modeling of this data takes into account the many aspects of this theoretical model, including the distance between the peaks, and that 4N populations should be twice as much as 2N populations, the CV, or spread of the peaks. And what happens in reality is that with biological samples, the CV of the peaks are wider. You can see here we're actually looking at a cell cycle analysis flow cytometry data file. The CVs are wider than, of course, 1 bin, as you might see in this theoretical model. And the 2N to 4N ratio might be slightly greater or less than 2. And you often end up with debris or artifacts within the data. And you can see here that we have some data that's kind of coming in here below the 2N population, as well as data that's coming in here above the 4N population. So what are these things, and how do we account for that with cell cycle modeling? Well, one of the common artifacts, or one of the common things that people see with cell cycle data with flow cytometers, is because cells do not always pass a laser in your flow cytometry one at a time. And you can see here in this little diagram that we have a cell passing the laser. That's a G1 cell, and you can see it's giving a certain signal peak, and that a G2 cell is also passing the laser and giving a peak that's twice as large. So this is our 4N and 2N populations. But what can happen is that a group of 2N cells or 1N cells or 1N and 2N cells stuck together can pass the laser, and you might be fooled into thinking that 2 G1 cells stuck together actually represents a G2 cell, or that 2 G2 cells stuck together might represent something greater than 4N. So what this means is these are generally referred to as doublets, and doublets don't just have to be two cells stuck together. Remember, doublets is just kind of a general term for any grouping of cells that are stuck together. And one way to perform a cell cycle experiment is to collect width, area, and height parameters, which can help you tease out single cells from doublets. Now, not everybody uses this technique, but it is one option to use. MultiCycle also has some sophisticated modeling tools for dealing with debris and aggregates that Kevin will talk about a little bit later as well. But you can see on this particular plot, we've highlighted what is less than the 2N population and greater than the 4N population. This data is currently ungated. But as we start to apply gates, like applying a scatter gate to remove anything that's not cells in FCS Express, you can see that these populations are already reduced. And there's still some populations here in less than the 1N, less than 2N, and greater than 4N. But if we start coming in and looking at our doublet discrimination parameters, the FL2 width and FL2 area, and FL2 height versus FL2 area, well, as we start applying these gates, we'll apply our area versus height gate in the hierarchy. You can see that this data gets cleaned up dramatically to remove these cells that are appearing as greater than 4N. So they're most likely doublets. And if we continue to apply the next gate in our hierarchy, our area versus width gate to this plot, it's subsequently cleaned up a little bit more. So again, using doublet discriminator parameters is a great way to kind of clean up your data. Again, it's not always necessary. It depends on what you want to do and how you want to perform that analysis. And again, MultiCycle has some very sophisticated tools for kind of teasing those populations out for you that we'll talk about later. Now with image cytometry data, as you can see here on the right, where we have a histogram displaying all of the RFP positive cells, which in this case are stained for DNA dye. They're represented in what's called a data grid down here. What can happen with image cytometry is that when segmentation routines mark two cells as one, or if they cut off a piece of a cell during segmentation, these can appear as plus 4N populations or lower than 2N populations. So what I'm going to do is apply this current marker, this image cytometry gate, to our data grid, and we can take a look at what these populations actually look like. So if we expand this marker out to the right, you can see in this case that these are somewhat segmentation artifacts. Here is an artifact where a very large cell is kind of being included in this routine. We're also kind of maxing out in some of our brightness here. So these are types of things that you may want to exclude from your image cytometry experiments for cell cycle data. And down on the other end, when we actually look at some of the cells that are here, you can see that in our image segmentation, or in our images that are being derived from this data, that the segmentation routine has cut off a piece of that cell. So it's actually not an entire cell, and you would probably want to exclude that for your analysis. So again, we can take these things into account by perhaps setting a gate for our image cytometry data to include what we know to be part of the model there, or as part of the data. We can also set up subsequent gates with image cytometry data, much like we do with the scatter gates and flow cytometry data, but using some of the other morphometric parameters. So again, that's a bit about the theory of working with some of the cell cycle data in FCS Express, or working with it in general. And I want to move on to talking about working with and inserting multi-cycle plots and data directly within FCS Express itself. So again, you can see we're in FCS Express. We have some DNA cell cycle results located in this particular layout that we're showing you here. And let's first start by talking about how to insert a multi-cycle graph before we talk about all the kind of beautiful formatting and reporting that we can do within the software. So to do this, we'll open up a new page. And the way that you insert a multi-cycle modeling or graph in FCS Express is to choose the multi-cycle plot from 1D plot options. We draw an object on the page, And if you have data loaded, you'll simply choose the parameter of interest that you want to load into that multi-cycle plot. If you're loading data from scratch, you'll be prompted to go find that data file and then choose the parameter of interest. But I'll choose FL2A. I know this is our multi-cycle, or this is our DNA analysis parameter. And you can see that this plot has been inserted. MultiCycle has already determined some of the modeling associated with this. You can see that some of the peaks here are highlighted in hatch marks in red and blue, and we'll talk a little bit about what these actually mean. You'll also notice that if you were to click on the plot in the Multicycle tab within FCS Express, these features now become available to you. The different models that can be used to display the data or model the data within Multicycle, which Kevin will talk about, as well as some of the fit parameters that are in there. Now you can see there's a number of different overlays or hatch marks or colored peaks, and let's discuss what these actually mean. We can double click on this plot to open up the formatting tab in FCS Express. And in the formatting tab there's an overlay section which will show you all the different overlays associated with this particular modeling or data plot. And let's actually move this over a little bit so we can view the overlays a bit more easily. And you can see that we have the original FCS file, which is this bd.lmd.fcs. This is the base overlay for these multi-cycle plots. We've also inserted overlays for noise, which Kevin will talk a little bit about with his description of multi-cycle, as well as the raw fit. And any of these overlays can be changed. So if we need to actually increase the line size for our fit, you can see that now on the plot, our actual fit is being highlighted a little bit more clearly. And we have separate overlays which can be controlled for their fill, their transparency, the color, the styles, for all the different models and populations that have been defined here. So in this case, MultiCycle is determining a G1 for diploid and for aneuploid populations. So all of the formatting can be handled there. You'll notice that MultiCycle did try and fit this data one way. When we inserted it, it went into the auto-fit mode to try and fit that data. There's quite a number of different fits that can be used for with MultiCycle in FCS Express, especially when you have the full version of MultiCycle. You can see here in this particular layout that we have a multi-cycle plot with a number of different data files. What I'd like to do is just jump between these data files to show you how the auto-fitting works. So you can see that as we skip between a different data file, MultiCycle is going to try and automatically fit that data for the appropriate cycle for that data file. And you can see here there's a number of auto-fit cycles that can be chosen or determined from that we'll talk about a little bit later. You can see here from the list of all these different things that are in there. So MultiCycle is going to do its best to try and fit the data as appropriately as possible for what it sees. Now you can also come in and manually format any of the MultiCycle plots if you like. So by default here we try and auto-fit something for you, but if you'd like, you can come in, you can turn off the auto-fit, and that will allow you to choose the cycle for these plot and also manually enter your fit parameters. So I can see with this plot it's determining to be a 2-cycle analysis, but I may want to force this to use a 1-cycle. I think it's a little bit more appropriate for this particular data file. And you can see here now that the aneuploid population that was highlighted in blue is no longer present because we're using a 1-cycle fit for this data. Now again, if we format this plot by double-clicking on it and coming into the formatting tab, there's a whole series of DNA options here in the DNA category for formatting these multi-cycle plots. You can see here in the General tab, we have the option to choose between any of the 6 different models to display on there. So models 1 through 6 correspond with manual, slice, G2, aggregates, S-phase, C-phase fix. Again, these are things that Kevin will probably discuss a little bit more in his portion. But importantly, you can come in and you can change manually how MultiCycle is trying to fit this data within FCS Express. So the first thing we had done with this data is we moved from a 2-cycle to a 1-cycle fit. We removed the automatic fit. This checkbox will control the little icon button up here as well. And you also have options if you need to change what you think the estimated CV for your peaks is going to be, or the spread. You can see here we determined a very kind of low CV of 2.5. If you needed to come in and manually change this, reduce it, or increase it, of course you could do so. You can also fix the G2 CV to the G1 CV so that MultiCycle is trying to fit based on similar CV values rather than doing that separately, as well as forcing where the G1 and G2 mean is to be determined. So if MultiCycle is not picking up exactly where you think the G1 mean or G2 mean is, you can come in and you can manually change that. Also, if you ever have data where your G2 to G1 ratio is not quite 2, or if it's a little more, a little less than 2, you can always come in here and manually check this option to fix a ratio. This can happen for a number of different reasons, but again, you can come in and make that change if necessary, as well as changing the order of S. And what the order of S means, there's 3 orders that you can choose from, 0, 1, or 2. And to highlight these orders of S, what I'd like to do is format this plot just a little bit so we can zoom in on the y-axis to view this S phase. And what the order of S means is that a 0 order of S assumes a kind of broadened rectangular shape for the S phase population within the MultiCycle plot. An order of 1 assumes a kind of broadened or tilted trapezoid. So if I click this, you can see that the S phase now looks to be decreasing. And if we choose an order of 2, this is going to give us an S phase that's kind of higher on the sides and lower and tilted in the middle. So again, all of these things can be changed. If you need to manually come in and make some adjustments for your data sets, for whatever reason, you can do so. One thing to keep in mind is that the defaults for MultiCycle and FCS Express are assuming the theoretical model that 2n is half as much as 4n. And the more changes that you kind of make here for manually fitting, the further away you're getting from the theoretical model, and the further away that your data sets are actually getting from what we're expecting. So keep that in mind as you're making any kind of manual adjustments here. Now, some of the other things that are included here is that we can actually show the position markers for any of these plots. So let me get ourselves back to a normal looking plot. So if you'd like to show where the G2 and G1 means are, you can simply pick to choose to show these position markers on the plot. We'll make these bright blue so we can see them a little bit easier. You can see that lines will be inserted where these G2 and G1 means are, which is a kind of convenient way to quickly show where everything is. Now, if we want to view some statistics from our MultiCycle plots, it's very quick and easy in FCS Express. You can simply right-click on your MultiCycle plot, and from the statistics category, you can choose 3 MultiCycle DNA statistics. So we have our standard DNA cell cycle statistics, we have our DNA experiment statistics, and we have our DNA model summary statistics. So again, I believe Kevin will be talking a little bit more about kind of what some of these statistics mean and what they do for you, but you will have available to you at a click, all of your percentages that the model has determined for, your different phases of the cell cycle. Now again, another nice thing about FCS Express when you're using MultiCycle here is that you can format these plots any way that you'd like for presentation, publication purposes, or even just to impress your peers or your colleagues. And again, all of this can be handled by double-clicking or right-clicking on the plot to choose format. And what a lot of people like to do is come into our overlays and just make these look a little bit nicer by adding maybe a solid fill and some transparency, maybe changing the colors of the different cell cycle phases. So here we'll change our diploid S to orange and our G2 to green. And when we do this, you can see that we're going to end up with a very nice looking multi-cycle plot where we've highlighted the different populations. We have some transparency going on. And this can be taken to the point where we generate really nice looking reports, kind of what we've done here on the first page. When these reports are created, when the statistics are inserted, you can simply move your gates, and you'll notice that our cell cycle statistics and the plots and the models will update accordingly when those gates are changed. We can also do some kind of fancy things in FCS Express, like using bar plots and pie charts to actually chart some of that data. Again, we have other kind of resources and webinars if you want to learn more about how to create these type of charts. But again, any of the gates that you create that can be applied directly to your multi-cycle plot. And conversely, if you'd like to actually create a gate on a multi-cycle plot, you can simply create a marker on that plot, convert that marker to a gate by right-clicking, and you convert and link that to a gate. So you can see here that my create a new gate window has appeared. Here's gate 1. And if I'd like to at this point, I can say here's my approximation of G2 cells for my marker, not from the model of course, so I'll apply it to this particular plot. And the nice thing is, as I update this, we can see in any of the plots where these populations fall on any other charts within FCS Express. So again, there's a lot of benefits to using multi-cycle, of course, for the sophisticated modeling. In conjunction with FCS Express, we can generate really nice reports and really get all the kind of statistics and things that you need. Now, in the end, when you're ready to export some of this data, you can simply come up here to the File tab and choose Export. We'll choose to export this as a PowerPoint presentation as an example, and we'll open this presentation. And what will happen is that PowerPoint will automatically launch, populate itself with your report, and then you're going to have a high-resolution report. All the graphics that come out are high-resolution vector graphics that can be moved and resized and moved around the page. So this is perfect if you need to grab something for publication, if you need to put together a poster, or if you need to kind of send a report to a friend. All of this can also be done within what we call the batch process in FCS Express. We won't be covering this today, but you never collect one sample or two samples. If you have 100 samples, we can simply choose to export all of those samples to PowerPoint to PDF to Excel with a click of our batch processing run button here. Again, we have lots of resources that can kind of cover those things on the Novo Software website if you'd like. So at this point, I wanted to open up the floor for a few questions while we transition over to Kevin. I'm going to bring Kevin on the line here. Hey, Kevin, you should be live with the audio.
Yes, I hear you loud and clear.
Great. I'm going to make you the presenter, and I'm going to take a look here and see if we've had any questions come in so far.
Welcome, everybody. My name is Kevin Becker from Phoenix Flow Systems. I'm dating myself when I say that I have been selling this product for close to 30 years, but flow cytometry is one of those infectious things that makes people stick around for a while. So I'm still here, and I'm still hanging with cell cycle analysis. Sean, do we have any idea of people? I don't see, are people like novices at cell cycle, or do we have any idea who we're talking to here or not?
I think we have a group of people that ranges from people who have never maybe done cell cycle at all to people who are doing it day to day. So I would probably gear this to the intermediate level of user, and assume we know nothing and everything at the same time.
Okay, and does anybody have a specific question about cell cycle that we can try and address? Are you getting any feedback on that? While you're typing in your answers, I just want to say, Sean, that Thanks. That was a really good overview. I learned quite a few things, and I'm not quite sure why you have me here, because you seem to have the whole thing mastered in FCS Express. And that was really helpful to me, learning about getting the vector graphics. So using PowerPoint, is that the best way to get high resolution graphics out of FCS Express? Exporting whatever it is on the screen to PowerPoint, that works better than the other modes in the program?
Yeah, there's quite a few options for exporting these things. You know, the kind of standard export to PowerPoint works quite well, but really anybody can just kind of right-click on a plot and choose to save that plot as a TIFF or a PNG or a JPEG. So there's a number of kind of variety of different things that people may want to use. But if you want to generate a kind of full report in PowerPoint, then of course export to PowerPoint is best for you. But if you really need kind of one super high resolution image, you can simply just do an export to TIFF or to PNG if needed.
Okay, thanks. Thanks for that clarification. As you can see on the screen, we just have a simple DNA histogram here. And so before we start talking about what all of this stuff means and what we can do with it, I'm just going to switch over here. This is what I like about FCS Express, is that we can put up multiple things at one time, not only just our numbers, but some points that we'd like to make. So did we get anybody that wanted to ask a specific question before we get started, Sean, or not? No?
I don't think we've had any specific questions coming in, but I'm sure we'll get a few by the time we get to the end of the webinar.
Okay. So I have here a couple points about doing cell cycle analysis, which I thought that I would just go over first, because the way that Sean gave his presentation, it looks really fun. It looks really easy, and it's very powerful in FCS Express. But as we well know, that we very often reality doesn't look as good as when we do one of these presentations. And so, but I think the number one message that I can stress about cell cycle analysis before you start cell cycle analysis is this point right here, which is garbage in, garbage out. And the way that you can get the best, most reliable, most reproducible numbers in any sort of cell cycle analysis is to prepare your samples as well as you can. And by preparing your samples, what I mean is making sure that the CVs of the samples are as narrow as possible, as small as possible, and that you do everything in your staining protocol to minimize the amount of debris and junk in your sample, and that you especially be sure that you have enough DNA dye in your sample so that you are staining all of the cells completely. And if you can do all of those things, that's 80% of the doing cell cycle analysis. If you have good data, then you're going to have good success with the cell cycle analysis software. And the other thing, another point that I wanted to make too, is that we do debris modeling in cell cycle. That's what this area is right here. Is that this is a mathematical calculation where the software is looking at how much of the sample has debris in it, or how much of the sample is debris, and it subtracts that from your overall percentages of your different parts of the cell cycle. And this is extremely critical. This was the downfall in the beginning of cell cycle analysis, is because your S phase is what you're very much interested in often, and you can see even by this picture, and then I can flip over. I have another page right here. If I go to page 3, and you can see that the debris, which is this gray shaded part here, is going through the whole entire histogram, and we're concerned about our S-phase percentages often. That's our most important result that we want to get, and if we don't compensate for our S-phase results, if we don't compensate for our debris, then we're going to have elevated S-phase results. And one of the big issues in DNA analysis is reproducibility between different samples. And the one way to increase the reproducibility of your S-phase measurements is to let the software apply its mathematical model to subtract the whatever debris component is in there each time, so that way it increases the accuracy of your S-phase measurements. Another thing I should mention is with all of the flow cytometers and now having 10,000 channels in their DNA histograms, in their histograms in general, lots and lots of channels aren't necessary for cell cycle analysis. You only have a certain amount of accuracy in cell cycle analysis, and so by increasing the number of channels, what you're doing is that you're just making your life more complicated or you're making it take more time to run your samples, because what you need a minimum number of cells to get an adequate representation of your DNA histogram. And if you have more channels, then you have more bins. You're dividing your data up into more bins, and that requires that you collect more data in order to get adequate data in each one of the bins so that you can do the modeling. So by 512 channels, 256, 512 channels, and 10,000 counts is more than adequate for doing cell cycle analysis. So turn the resolution down on those histograms, and then you'll be able to go home a little sooner if you're collecting all of the data. Another issue here, which I wanted to talk a little bit about, is people often want to use cell cycle analysis to detect apoptotic peaks, because quite often people, so let me go here to shoulder phase. So here I have a layout here where I have loaded in some data here. And so quite often we have a simple DNA histogram here, and it has a shoulder on it. And they say, well, this is my apoptotic cells. Now I'm going to use the cell cycle software to determine what percentage apoptosis that I have. And then they attempt to do an analysis, and, well, they look at it and they say, well, that's 2 peaks. So therefore I should be able to model that. Wrong. Cell cycle analysis software is a tool, but it has limitations in it. And one of the rules of cell cycle analysis is, if you're going to do some sort of a analysis like this on this peak, and you want to separate it into 2 separate peaks, there needs to be some sort of a valley in between here. You just can't have a shoulder here, and you can't expect the software to be able to deconvolute this into 2 separate peaks. And so all that you can get from cell cycle analysis when you're looking at apoptosis like this is, yes, I do have some apoptosis, but I need to go to another assay, an annexin 5, a tunnel assay, some other assay where I can do a better job of pulling out my apoptotic cells and getting a measurement of what the percentage is. So don't be looking to cell cycle analysis as your solve. I know it's fast to do apoptosis that way, and it's cheap, but it's not reliable. Now, maybe we should go back here, and I just want to mention how the math. I think that Sean mentioned how the model is working, but what it is what it's trying to do is it's trying to apply a Gaussian curve to each one of the individual compartments of the cell cycle, and it's called the polynomial S-fit method, and it's based on a mathematical model that Dean and Jett invented about 45 years ago or so. They came up with this model, and I don't mean to slam any other softwares or anything, but I'll say that ModFit uses the correct model, and all of their math and everything, and the way that they do it is ModFit is an equally good program. But if any of your friends are working with FlowJo and the basic model of cell cycle that's in FlowJo, if they get really different results from you, the reason why is because they aren't using this model to model their data. So let's look at a couple files here and see what we have. So just for some analysis. So let's take a look at, and then let's go over what the different things that. So what I'm going to do is I'm going to go and I'm actually going to. So when I want to load a data file in here, I'm going to go here and I'm going to go to select a data file. And I'm going to, I should mention too, another function that, so as Sean says, MultiCycle can analyze up to 3 cell cycles. And I mentioned the fact that when you had 2 peaks that were close together, such as an apoptotic peak and stuff, it was really, just a shoulder didn't work. And so what we have on this one right here is we have 3 cell cycles. And if you look at it, it's kind of hard to tell, but you see that there's a dip here in between the two, the peaks, and we have a little bit of a dip. But so we have 2 overlapping aneuploid populations. And MultiCycle can analyze this. And so what we do here is we go to MultiCycle, but as he mentioned before, MultiCycle comes up into an auto-fit mode. And auto-fit, it can't figure this out, so you need to help it out, and so to make a decision. And so what we do here is we click on fit parameters and we tell it, we turn off the auto fit, and then we click on this and then we say this is 2 cell cycles. The auto fit is not going to always work all the time, and then in the case of this, what we also want to do is, because one of those, we want to say that those 2 CVs are equal, the CVs of all of the peaks are equal because they so overlap so much, and then we hit OK, and then what happens is MultiCycle has just divided this in half so that it looks like, that it appears that it's has fitted both of these, but in reality this is a bit of a stretch. So that's why cell cycle analysis software is a tool, and it requires some experience in using it when you start getting into these complicated things, and you as the user have to decide whether the results are believable or not. But we do try to help you in the software, because we do offer an interpretation over here. And what I just did was I flipped to a third page, and over here I have page 1 was full scale. Page 3 is I have increased the y-axis scale so that I can see the different compartments. This is a handy way to try to, this is one of the beauties of FCS Express, is you can set this up like this without having to go and changing the scaling on the of the y-axis. And so what it does here, what MultiCycle can do is it can offer some suggestions. It has a decision tree in it, and it can offer some suggestions on how believable the data is. But in this case, you really have to use your common sense, because you know that these were 2 aneuploid populations, and this area right here is the S phase of those 2 aneuploid populations. So any number that MultiCycle is reporting out here is going to have to be an average. And so in that respect, it shows you right down here that the average percent S of the aneuploid is 20%. So if you were trying to determine what the S phase was in this one, you would be remiss in saying that you could say what the individual S phases were, which it tries to report down here, and so instead you would want to report out an aneuploid, average aneuploid S phase. And so it's very much operator dependent. So let's look at another file here to see some of these other features that MultiCycle has in here. So let's look at, I can't remember what this one is. Let's take a look at this one. Okay, first let's go back to here and see. Okay, so what we have here is, looks like it didn't do a very good, what it did on this one was, it looked at it and it did a quick fit and it didn't get it right at all. And so we need to help it out on here. And once again, so we go up here to here, and we need to tell it that click on here, and let's see, we go to fit parameters. Oh, it's still stuck in, oh, I see, it's auto detect. So let's see here now and see if see if that helps it. Oh, there we go. So what had happened on the last one was I had turned it manually to detect 3 cell cycles, and then I loaded another data file, and it had used I had forced it to say 3 cell cycles, and now this one wasn't 3 cell cycles, and so now I put it back on auto, and so it was able to detect that this was 2 cell cycles. And what we have, so now let's go over here to, what you have here is, going across here, you have this thing that says Model 1, Model 2, Model 3, Model 4, Model 5, Model 6. Remember we told you that, or I told you that it tries to give you some sort of a suggestion about how well the model is fitting the data. And it's, you know, on this one it says the S phase confidence is good. And how it makes this decision is that it automatically, for what it does is it automatically varies, I'll move this over here, it varies different parameters on the cell cycle, and the theory is if it changes the parameters on the cell cycle and it gets a huge shift in the results, then that means that it's having a hard time fitting the data, whereas if it changes certain parameters and the data is fairly accurately together, the results are similar, then that means it's doing a better job of fitting the data, and that means that your results are more believable. And so when we have this models up here, these are the 6 different ways that it's automatically fitting the data to come up with this interpretation. And so model 1 corresponds, you see that now suddenly that the S phase went from being, it was order of 1, and then it went from being order of 0 here. And so if you see over here, this is what I put this up here, and this is the information from the 6 models. And so model 1 corresponds to this one right here, model 2 vice versa. And it's a little bit of a secret code that you have to learn, but this means S0 means slice nuclei for the debris, and the S phase is 0. Slice nuclei clumping, the S phase is 0. The G2, G1 phase is fixed. Apply the aggregates model, which is something that we haven't talked about yet. The S order is equal to 1, which means it's sloped, and this one means that the CVs are fixed of all of the histograms. It means that they're all the same. So what you have to do is, what you can do, so by pushing this button here, you can graphically display the results of each one of these different models. Now, you say, oh well, this is all great, but which one is the right one to use? Which one are the results are the best or the correct? What is, or how am I fitting the data the best? And that partially comes from experience, but also from looking at the data. And because, so what you're going to have to do is that you're going to have to look at the raw data, which in this case is by the line, by this black line that's along here. Normally the S phase is where you look this is where you see the most variability, and you have to just, if you're really trying to be precise about this, you, instead of letting the software pick the model, then you have to look at each one of these and decide which one you think fits the data the best. But if you look over here at the statistics, you see that the numbers don't change that much. Like our let's look at right here, our aneuploid %S. So we have 20%, 17%, 22, 18, 20, 21. So we don't have a lot of variability in there. So what we can do to try to make the decision is that we can look at the chi-square. And this is another statistic that's in CellCycle, which is provided by it. And this goes back to one of my other points, which was one of the other points is, what is a good chi-square value? A good chi value, and so chi-square is a measure of how well the mathematical DNA cell cycle model is fitting the data. So that means we don't analyze our data so that we go for a higher or a lower chi-square. What we do is we fit our data so that, well, actually I said that wrong. We do go to try to get the lowest chi-square is what we try to do, because we are trying to make our mathematical model fit the data as well as possible. But in certain cases, you don't apply an arbitrary number. By that I don't mean you don't always say, I got to get a chi-square of 2, otherwise my data is not, that I got is not reliable. The whole idea is if I get a chi-square of 10, as we had on here, we had a range of chi-square from 3.7 or 4.6 to 2.7, and so the lowest chi-square would be in this model right here, which is sliced debris modeling, clumping on, and the S phase is equal to 0. And then if I click up here, this is model 2, this is what my data looks like, and based on going for the lowest chi-square, then I now have done my best analysis of my cell cycle data. And see, here is the clumping that we talked about right here. This is part of the noise function right here, and so this is saying that this part of the data, this is the noise function. It goes along here, and then it goes to the debris up there, and that's part of the, it's too small to see, you can't see it here, and it's subtracting all of that from your cell cycle. And so those would be your correct cell cycle results to report for this particular histogram based on using the chi-square as a criteria. Or once again, MultiCycle says the S phase confidence is good and that an aneuploid DNA population is observed. So it gives you a few hints, because you've got to use knowledge, your own knowledge, to interpret the results of cell cycle. Sometimes it's not clear cut. So does anybody have any questions at this point? Is anybody still awake at this point?
I'm still awake.
I'm getting
Well, thanks, Kevin. Yeah, are there any questions anybody has for Kevin at this point? We're getting some responses that other people are awake as well, and they've been watching.
Okay.
So one of the earlier questions that came in was essentially, is there a fitting algorithm for sub G1 analysis? I think you had covered that, discussing about the apoptotic peaks and why those can't be found. Are there any plans to include any sort of sub G1 analysis in the future? Is that something that you think you'd like to see, or something that we should stay away from?
There is an option to, there is an option in the software to fit a peak. There is an option to, if you have a defined peak, you can, in the software here, you can fit a, if you have a standard or an asymptotic peak, you can fit it, or you can fit it as a single peak. You can do that, but the issue always is it seems only about 10% of the people have that success, and the rest of the people have shoulders.
Great. And we just had a question about what is the green population in this graph that's up right now?
Okay, so this is something that one of the this is one of the options in MultiCycle, which, if you need to use it, is a very handy model, but not very many people are aware of it. Okay, and that is, MultiCycle has the ability. This is called a synchronous S analysis, and so what has happened is that the cells have been arrested in S phase, and so we have all of our cells all bunched up in S phase, and so what it is it's based on Fox, Michael Fox. He's not around anymore. He came up with this idea that you could model the arrested S as another Gaussian peak inside the S phase, and so this was added to MultiCycle. Not so many people use it anymore, but this is another, in addition to all of being able to analyze all the aneuploid peaks and to analyze an extra peak. You can model as a synchronous. This is called a synchronous S DNA histogram.
Great. So I don't see any other questions coming through right now. What I'd like to do is just flip the presentation back to myself. Maybe we'll have a few more questions come in before we sign off here. And I just wanted to give a little summary of kind of some of the things we talked about in that basic cell cycle analysis with MultiCycle plugin. That's going to come for free with FCS Express flow and image cytometry. And any of the kind of more advanced cycles that you may be needed, those can be purchased as an add-on to FCS Express. We had a question about that come in. You can find out the pricing for that on our website at novosoftware.com. Any of the plotting, the data analysis, the mathematical modeling, all the advanced report generation tools, they're all in one package now, which makes everything very convenient for doing this analysis in FCS Express. While FCS Express Image is going to leverage all of the really powerful and proven tools of flow cytometry, including this MultiCycle cell cycle DNA analysis, which is really cool that the flow and image cytometry data work essentially the same way, as long as you can collect enough events from your image cytometry data to make statistically significant populations in the graphs. Novo Software also provides many resources, like the webinar we're holding today, including tutorials and sample data. There's a whole tutorial on cell cycle analysis using MultiCycle on the Novo Software website. We also encourage you to visit the Phoenix Flow Systems website at the web address down here, and you can address any questions for Phoenix Flow System at their website, or you can write us to support@denovosoftware.com. And I see a few other questions coming in here. We'll hang on the line for a few minutes to talk about those. And thanks again for everybody for joining us today on the webinar.